All right, well, welcome to, let me switch this around. Lesson 19.3, Earth's early history. This is probably pretty interesting and fascinating for most of you. The goal is to describe the scientific explanations of the origin of life on Earth. And the far side says, nothing yet. How about you, Newton? Okay. So our objectives are to identify some of the hypotheses about early Earth and the origin of life, also to explain the Endosymbiotic Theory, and explain the significance of sexual reproduction in evolution. So you probably have thought about it. What's going on here? Oh, there we go. Here we go. How did life on Earth begin? What were the earliest forms of life, and how did the life and the biosphere interact? The origin of life research is a dynamic field, but even though some of the current hypotheses will likely change, our understanding of other aspects of the story is still growing every day. So objective 1 is to identify some of the hypotheses about early Earth and the origin of life. I tried to find some interesting pictures for you guys to look at, obviously drawings. Geological and astronomical evidence, sorry, it's another late night, suggests that the Earth formed as pieces of cosmic debris collided with one another. While the planet was young, it was struck by one or more huge objects and the entire globe melted. For millions of years, violent volcanic activity shook the Earth's crust. Comets and asteroids bombarded the surface. Then, about 4.2 billion years ago, Earth cooled enough to allow solid rocks to form and water to condense and fall as rain. Earth's surface became stable enough for permanent oceans to form, and this infant planet was very different from what we know of Earth today. Wow. Earth's early atmosphere contained little or no oxygen. It was principally composed of carbon dioxide, water vapor, and nitrogen, with lesser amounts of carbon monoxide, hydrogen sulfide, and hydrogen cyanide. Not a very nice place, though, huh? Because of the gases in the atmosphere, the sky was probably like a pinkish orange. And because they contained lots of dissolved iron, the oceans were probably brown. So in 1953, chemists Stanley Miller and Harold Urey tried recreating conditions on early Earth to see if organic molecules could be assembled under these conditions. They filled a sterile flask, oh my gosh, this is terrible, with water to simulate the oceans and then boiled it. To the water vapor, they added methane, ammonia, and hydrogen to simulate what they thought had been the composition of the Earth's early atmosphere. They passed the gases through electrodes to simulate lightning. Then they passed the gases through a condensing chamber, as you can see right here, where cold water cooled them, causing these drops to form. All right. The liquid continued to circulate through the experimental apparatus for a week. Then after the week, they had produced 21 amino acids, which, as we know, are the building blocks of proteins. Miller and Urey's experiments suggested how mixtures of the organic compounds necessary for life could have arisen from simpler compounds on primitive Earth. We now know that Miller and Urey's ideas of the composition of the early atmosphere were incorrect. Their experiment used water, methane, ammonia, and hydrogen. There is now abundance of evidence of major volcanic eruptions 4 billion years ago, which would have released carbon dioxide, nitrogen, hydrogen sulfide, and sulfur dioxide into the atmosphere. But new experiments based on current ideas of early atmosphere have produced similar results too. So geological evidence suggests that during the Archean eon, 200 to 300 million years after Earth cooled enough to carry liquid water, cells similar to bacteria were common. How do these cells originate, though? Where do they come from? Large organic molecules form tiny bubbles called protenoids, microspheres, under certain conditions. Microspheres, as you can see here, are not cells, but they have some characteristics of living cells. Although they do reproduce asexually by bonding, they do not pass on any type of genetic material. However, they may have been important in the development of life, providing a membrane-enclosed volume, which is similar to that of a cell. So they were enclosed in a membrane. Microspheres, like cells, can grow and contain a double membrane, all right, which became more complex. So they could actually carry on lifelike functions. These double membranes can actually diffuse the materials and cause osmosis for water to go across them. They would eventually become heterotrophs, organisms with the ability to absorb nutrients from the environment for the energy and growth. And as the amount of nutrients in the environment decreased, competition for these precious resources then increased. Heterotrophs with more complex biochemical reactions would have an advantage in this competition. Several hypotheses suggest that structures similar to proteinoid microspheres acquired the characteristics of living cells as early as 3.8 billion years ago. Cells are controlled, as we know, by the information stored in DNA, which is then transcribed into RNA and then translated again into proteins. The RNA world hypothesis about the origin of life suggests that RNA evolved before DNA. From this simple RNA-based system, several steps could have led to DNA-directed protein synthesis. A number of experiments that simulated conditions on early Earth, such as small sequences of RNA, could have formed from simpler molecules. Under the right conditions, some RNA sequences help DNA replicate. Other RNA sequences process messenger RNA after transcription, as we know. Still, other RNA sequences catalyze chemical reactions. Some RNA molecules even grow and replicate on their own. Microscopic fossils, or microfossils, are prokaryotes that resemble bacteria, have been found in Archean rocks at more than 3.5 billion years ago. Those first forms of life forms evolved in the absence of oxygen because at that time Earth's atmosphere contained very little of that highly reactive gas. The pink spots you see on the map here are the Archean rocks where these microscopic fossils were found. Then later on, during the early Proterozoic eon, photosynthetic bacteria became common. This is a couple of examples of photosynthetic bacteria. By 2.2 billion years ago, these organisms were producing oxygen. So at first, the oxygen combined with the iron in the oceans, producing iron oxide, or rust. Iron oxide, which is not soluble in water, sank to the ocean floor and formed great bands of iron that are the source of most of the iron that we mine today. Without iron, again, because the oceans were brown, because of the iron, without the iron now, the oceans change color from brown to a blue-green. Next, oxygen gas began to accumulate in the atmosphere. The ozone layer began to form, and the skies turned to their present shade of blue. Over several hundred million years, oxygen concentration rose until they reached today's levels. Many scientists think that Earth's early atmosphere may have been similar to the gases released by volcanoes today. The graphs show the composition of the atmosphere today and the composition of gases released by a volcano. Here's volcano, and here's atmosphere today. Lots of nitrogen, oxygen, other gases. Water vapor, carbon dioxide, nitrogen, and a lot of other gases too. To the first cells, which evolved in the absence of oxygen, this reactive oxygen gas was a deadly poison that drove this type of early life to extinction. Some organisms, however, evolved new metabolic pathways that used oxygen for respiration and also evolved ways to protect themselves from oxygen's powerful reactive abilities. Objective 1 was to identify some of the hypotheses about early Earth and the origin of life. Early Earth's atmosphere contained little or no oxygen. It was principally carbon dioxide, water vapor, and nitrogen, with lesser amounts of carbon monoxide, hydrogen sulfide, and hydrogen cyanide. Miller and Urey's experiment suggested that how mixtures of inorganic compounds necessary for life could have arisen from simpler compounds on a primitive Earth. Finally, the RNA world hypothesis proposes that RNA existed by itself before DNA. And from this simple RNA-based system, several steps could have led to the DNA-directed protein synthesis. So let's move on to the endosymbiotic theory, which we've talked about before in this class several times. So one of the most important events in the history of life was the evolution of the eukaryotic cells from prokaryotic cells. Eukaryotic cells have nuclei, but of course prokaryotic cells do not, as you know. Eukaryotic cells also have complex organelles. Virtually all eukaryotics have mitochondria, and both plants and green algae also have chloroplast. So it is believed that about 2 billion years ago, some ancient prokaryotes began evolving internal cell membranes. These prokaryotes were the ancestors of eukaryotic organisms. All right, internal cell membranes started to evolve. According to the endosymbiotic theory, then, prokaryotic cells enter those ancestral eukaryotics, as you can see right here. The small prokaryotes began living inside the larger ones. Over time, a symbiotic relationship evolved between the primitive and eukaryotic cells and prokaryotic cells in them. The endosymbiotic theory was proposed more than a century ago. So at that time, microscopists saw that the membranes of mitochondria and the chloroplast resembled the cell membranes of free-living prokaryotes. This observation led to two related hypotheses. One hypothesis proposed that mitochondria evolved from endosymbiotic prokaryotes that were able to use oxygen to generate energy-rich ATP molecules. Without this ability to metabolize oxygen, cells would have been killed by the free oxygen in the atmosphere. Another hypothesis proposes that chloroplasts evolved from endosymbiotic. Well, I missed a spot here. Let's go back. Where are you? Did I go back? There we go, sorry. Prokaryotes that had the ability to photosynthesize. All right, so over time, these photosynthetic prokaryotes evolved within the eukaryotic cells into what we know as chloroplast of plants and algae. Pretty simple, actually. So, I missed something there. Objective 2 is explain the endosymbiotic theory. The endosymbiotic theory proposes that a symbiotic relationship evolved over time with primitive eukaryotic cells and the prokaryotic cells within them. All right, objective 3. Explain the significance of sexual reproduction and evolution. You kind of know all this stuff already, I think. So when prokaryotes reproduce asexually, they duplicate their genetic material and pass it on to daughter cells. This process is highly efficient, but it yields daughter cells whose genomes duplicate their parents' genome. So genetic variation is basically restricted to mutations in the DNA. When eukaryotic cells reproduce sexually, offspring receive genetic material from two parents. Meiosis and fertilization shuffle and reshuffle the genes, generating lots of genetic diversity, as we learned during meiosis. The offspring of sexually reproducing organisms are never identical to either their parents or their siblings, except, of course, for identical twins. Genetic variation increases the likelihood of populations adapting to new or changing environmental conditions. Multicellular organisms evolved a few hundred million years after the evolution of sexual reproduction. Early multicellular organisms likely underwent a series of adaptive radiation, resulting in greater diversity. So, explain the significance of sexual reproduction and evolution. The development of sexual reproduction sped up evolutionary change because sexual reproduction increases genetic variation. Our objectives, again, identify some of the hypotheses about early Earth and the origin of life, explain the endosymbiotic theory, and explain the significance of sexual reproduction and evolution. And all those things definitely support our goal of describing the scientific explanations of the origin of life on Earth. Dun, dun, dun. There's a cutie. Enjoy.